Okay. Good morning, everyone. Welcome back to day 5 of the 10-day workshop on high-end uh, workshop on computational biology and data science, sponsored by Accelerate Vigyan, Department of Science and Technology, Government of India. It's a great pleasure for me to introduce the speaker of the day, Dr. Sonika Tyagi from Monash University, Australia. Dr. Tyagi is a bioinformatician and a computational biologist who obtained her advanced diploma in bioinformatics and her PhD in computational biology and bioinformatics from Jawaharlal Nehru University, New Delhi. During her PhD, she received a Research Exchange Award to visit the University of Queensland in Australia, where she worked with Professor Tim Daly at the Institute of for Molecular Biosciences. From November 2010 onwards, she worked for the Australian Prostate Cancer Research Centre, Queensland, as a biostatistician. She is currently a group leader for bioinformatics at Monash University. Her research focus is on developing computational methods to retrieve knowledge from vast amounts of data being generated using high throughput techniques and biological experiments. She has developed many bioinformatics tools and applications using machine learning, data mining, and statistical approaches. These softwares are open source and available for users via free web interfaces. With that, I welcome Dr. Sonika Tyagi to give her talk on open source tools for genomics. Thank you. Thank you for coming here.
Thank you, Raghu, for a nice introduction. And good morning, everyone. It's afternoon for me. It's around 2:40 p.m. here in Melbourne. Great. So it's a smallish group. Anyway, I had created an Etherpad. I'm pasting the link in the chat. And I would like to just know my audience a little bit. So if you can fill out your name, where you are from, and maybe some keywords about the research you're doing, or if you're a student, just write student and what course you're doing. So that will give me an idea who my audience is. And then we can start. Yeah, so while you're doing that, yes, I work at Monash and I teach as well as do research. And today I'm going to share some of the work that we are doing, and I'll just give a brief introduction to some general open source tool and some bioinformatics specific tools. And I'd like to keep the talk interactive, and yeah, feel free to stop me and ask questions. Thank you. I'll start sharing my screen. And if someone can confirm that you actually see the full screen view and not the speaker's view.
Yeah, we can see your slides, Sonika. Yeah.
Full screen, right?
Yeah.
Awesome. All right. So this is how I had broken down our 1.5 hour just to get an idea, how much time I'm going to spend on different topics. So I'll just give you a brief introduction to what my group is working on. And then what we are not covering in this session is that we're not going to do any hands-on activity. I'm only going to introduce you to various topics, and we will not go into fine details of the algorithms because I'm aware that you're also doing hands-on sessions after these talks. So yeah, that's where probably you'll be learning a bit more about, things that you can do on your own with the data. Okay, so let's start. So the focus areas of my lab can be divided into these 3 aspects. So we are looking at transcriptomic study. We look at both coding and non-coding genes in there. We look at epigenomics. So this is the information on top of genomic information where we have regulatory elements. We have non-coding RNA and variants of those regulatory elements and how they affect gene regulation. And in the end, we are then looking at how to put all of this information together that's coming out from various gene level, protein level, it could be regulatory or metabolome level, and how to put this all together to get a system genomics view of any system. So to begin with, again, same thing. I'm going to introduce you to some three main projects that are currently going on. So the first one is that we developed a model to identify microRNA secondary structure. This is a project that started during my PhD, and I've been making lots of updates to that method that we had developed. So the method is called CID-miRNA, and it's based on a computational algorithm called SCFG, or Stochastic Context-Free Grammar. So where we know that microRNA are formed by folding onto itself in a stem loop kind of structure, and we build a model to capture those interactions. And then this model was then developed as a web server where you can paste a sequence and can determine whether this sequence can fold into a stem loop like structures similar to microRNA. I had a student, Alex, and a group of year 11 and 12 students who came and worked with me and have also contributed to this project. So it's a nice story for me to share with you all that, if students are given some real life projects, real life problems to work on, they can do some amazing things. And this group of students also came with me and presented their work at a National Genomics Conference, which had never happened before in the life of this conference. Okay, so so this tool was originally developed for human, but has been made more generic since and has been applied on various other systems, mouse and bacteria as well. Now from there, I'm moving to look at long non-coding RNA. So long non-coding RNA, as opposed to microRNA that are only 22 nucleotide long and their primary transcript is around 150 base pair long. These RNA are much longer. They are longer than 200 base pair, and they can be located. So this the arrow in pink represents my long non-coding RNA, and the ones in greens are the coding transcripts. So these RNA can be located either in the intergenic region, or they can be antisense to a coding gene, or they could be transcribed by the same promoter, but in different directions. Or sometimes they are just embedded inside a coding transcript. So they can be located in various different configuration. And if you look at their function, they function by interacting with other proteins or DNA or other RNA. Okay, so they can act as a scaffold where they will provide binding site for other proteins and can make two distantly located elements on DNA interact with each other. Or they can act as a sponge for a microRNA and then affect the gene regulation. They can also directly interact with the messenger RNA. And when they interact with DNA, they can also form more tertiary structures such as R loop and triple helix. So this shows that not only their origin is very varied, they also function in many different ways. Now, slowly we are able to annotate more and more long non-coding RNA, but we recently did a survey of existing databases and tools that have been used to study long non-coding RNA. And we realized that there is very less consistency between different long non-coding RNA databases. We realize that a lot of these annotations may not be accurate, or there hasn't been a standard criteria to define the expression or the identification of these RNA. So that's why we decided to look a bit deeper into this field, and we came up with a method where we are able to identify these long non-coding RNA. So we've just published a preprint today where we explain the detailed methodology of this tool. So this tool works on a deep learning model where we are able to identify as well as annotate these long non-coding RNA, and hopefully in future this tool can help accurately annotate these RNA. Okay, from there, I'll give you a brief introduction to the work that we are doing in the regulatory element detection area. So again, we have a pipeline that we developed in the lab. This pipeline is to detect transcription factor binding sites. So I don't know, from your coursework, you may be aware that genes are regulated by these transcription factors. They bind into promoter region mainly, and then they regulate expression of a gene. So we developed a model that we were using to identify these sites. So this is a cartoon diagram which shows that this is DNA, and where two transcription factor will bind in a sequence specific manner, and they recognize a particular motif to bind to. Okay, and then generally speaking, there is more than one transcription factor. They work with each other to regulate the expression of a gene. And then if we zoom into these motifs, so these motifs are just a consensus representation of various binding sites. So what I'm trying to tell you is that these proteins do not recognize just one type of sequence. So the same protein can bind to all of these patterns. Okay, so the binding site allows some variation, and the consensus representation of this binding site is known as a binding motif here. Okay, now these binding sites are very, very small. They're only like 5 to 12 base pair in length. And if you take an example of human genome, it's billions of base pair long. Okay, so there's a high chance if when you develop a computational model to find these sites, you end up getting lots of false positives. And if you just purely search the entire genome, you will get millions of binding sites for these transcription factors, but only few thousands of them will be actually active. So the challenge was to identify between these true sites from the false sites. So then we are further building on our existing pipeline to include this information that we are not looking at binding site of this protein in isolation. We'll look at collaborative motifs. So this is where we have developed, again, a machine learning based model where we are looking at co-occurrence of these motifs. And this model works based on an ANN architecture, and there are some of the details of the deep learning model that was employed. And we were able to develop a new model, as well as we have applied this to 231 known human transcription factors from a ChIP-seq database, and those annotated clusters of motifs is also available through a web-enabled database for other people to access. And we are in the process of annotating one more ChIP-seq database called ChIP Atlas to give more comprehensive annotation of all human transcription factors. And then we plan to extend this work to look at RNA binding proteins next. Now, so till this point, we've been looking at data that may be coming off these various technologies. So all the techniques that you see here that end up in a term called seq are various sequencing technologies. So for example, in ChIP-seq, you will be sequencing DNA that binds to a protein. In RIP-seq, you'll be sequencing an RNA that binds to a protein. Similarly, you may have data that represents chromosome conformation. You may have data that comes from RNA and DNA interactions. So we have now all these high throughput sequencing technologies are available that can capture information from these various parts of this, this chromatin, okay? Now, so this is the information we are looking at, purely at the DNA level, the regulation level, and then you could also be looking at information at the functional level, where you are sequencing protein or looking at the spectra of proteins, or you may be looking at the signatures of metabolites. Okay, so but when you are studying a complex condition, could be like a disease such as cancer. So it's not when you're studying. Trying to understand that system, you need to put all of these layers together, and this is what we are trying to do when we are studying a complex biological system. We want to be able to integrate any of these data that's available. So we call it a multi-omics integration approach, and this is where the major focus of our lab currently is. And again, we started looking into various multi-omics data integration strategies. And then we realized different people mean different things when they use the word integration. So first of all, in this paper, first we tried to tease apart, various ways people have used this word integration. So we divided up all those methods into two main categories. So one is intermodal. So modal means one type of data. Okay, so we are looking at two different type of data. One may be RNA-seq, one may be ChIP-seq. So this is two modalities of data. And another area where we found people have used the term integration is intramodal. So it could be just all RNA-seq data sets, but done in different batches, different experiment, or different technology, but it's all RNA-seq data. So we call it intramodal. So after separating our existing methods into these two major categories, we realized that our work where we are focusing is intramodal, where we can look at different modality of omics data and look at their integration. And again, we further subdivided into generic categories where the computational model that we're developing to integrate this doesn't bother whether data is coming from ChIP-seq or RNA-seq or exome-seq or genome sequencing, and we are able to integrate that data. The other group of methods are where they have very, very defined categories that they can integrate. Okay, so we came up with all these various kind of approaches that have been used so far, and we have implemented last two of these in the model that we are currently, the paper is under review, and we applied that to multi-omics data from COVID-19 condition, and we demonstrated that by using a generic intermodal integration approach, we are able to identify signatures that were not possible if you were studying one type of data separately. And further, we changed the term integration to harmonization in this case, and we say we harmonize multimodal data by using these machine learning and other computational approaches. Okay, and towards the end, we are planning to develop a universal data harmonizer. So this harmonizer does not mind, what type of data I'm putting into it. In the end, it's able to convert that into a format that can be compared between multiple types of data. Okay, so that was just a brief summary. Sorry, I did not give a lot of details, but hopefully you get an idea of various kind of projects that are going on. Does anyone have any questions before we move on? If you're if you're typing into chat, I may not be able to keep track of chat as I'm sharing my screen. Okay, there's nothing in the chat. Any questions, anyone? Okay. Okay, so let's jump into the main topic of today, which is about various open source tools. So I'm very, very thankful to the open science community where you are developing open source tool. They are open access. You can use them, reuse them, and modify them. And my entire research thrives on the availability of open research data as well as these open tools. So none of my work would be possible if these tools were not available for free. And I'm going to share some of these tools with you today. And that's also the reason why, when I'm developing some research software, those are again open access and open source. So some of the tools that I'm going to introduce are RStudio, R Notebook. So has anyone used R? You can unmute yourself and tell me, has anyone used R in this group? Don't be shy. Yes, one person. Thank you, Sangeeta. Anyone else?
Yes, ma'am.
Beautiful, beautiful. So you're some of you are aware of R. You can okay, no problem. I was going to say something else, but has anyone used RStudio? How do you use R? Through RStudio or through command line? Is it possible for you guys, whoever can turn their camera on? That'll be good. I'll be happy to look at my audience and not just stare at this my own slides the whole time. If you are able to, if you can, please turn on your camera. I love to see my audience when I'm giving a talk.
I request the attendees to not type it in the chat or Q&A. Most of you are typing yes ma'am through RStudio. Rather unmute and then reply to Dr. Tyagi directly. That would be great. Thank you.
Yeah, it's like, let's try and simulate the real classroom environment where, I could look at you and, you can answer me and yeah, let's have a bit of two-way chat. Okay. Okay.
Can you hear me, Sonika?
Yes, yes, Pulkit.
Great.
Okay, cool. Good. So it looks like many of you are using R and RStudio, and then another programming language that's very popular in bioinformatics is Python. So like RStudio is for R, there is Jupyter Notebook and Google Colaboratory for Python. So if you're doing Python programming, you may have come across with these two tools that you would use to write the code and execute it in a graphical user interface. Okay, so R is a programming language, it's a statistical programming language, but RStudio is an IDE, integrated development environment, where you instead of running R on command line, you could run it in a nice software. So RStudio is the name of a company who built this software. And then R Notebook is a way to write a code and documentation together. So our Notebook is yet another format where you can write nice reports in an interactive fashion. TensorFlow is a library of various programs that you would be using for machine learning. Okay, now on the right-hand side, I have some different categories of various bioinformatics tools that you will find in the open source area. So you'd have various aligners, you will have various assemblers, or you may have tools for specific type of analysis. For example, you may want to do gene expression analysis from RNA-seq data. You may want to look at variants, what are the parts of DNA are different to its reference genome. You may want to find new genes on a newly sequenced genome, or you may want to compare two sequences from two organisms or same gene from within with an organism to look at their conservation, to do more functional analysis. So there are tools specialized for all of those functions. I forgot to mention bioconductor here, but bioconductor, if you're doing programming in R for bioinformatics, you may have come across bioconductor already. There are more than 2,000 freely available packages that are available for bioinformatics analysis. Okay. Now a bit of history of R. So R was developed here in New Zealand by Rose Ihaka and Robert Gentleman. So these two people, they worked at University of Auckland in New Zealand, and they came up with this statistical programming language called R, and the R gets its name from their initials. Okay, so why advocating for R? R is free, it's portable, you can run it on any system. As I said, there's a huge community out there who are constantly developing and maintaining all these packages related to just, it could be just general, data science concepts, or they may be specialized function that these packages have been developed for. And I saw in your program schedule for this afternoon that you'll be using one of those packages to do some analysis in the hands-on session. R is becoming very, very popular language in various research streams. So anything to do with data, you will find R-based analysis there. And there's some wonderful graphics methods that are available through R. You can really develop nice, publishable images using this language. And all of these big companies, be it Twitter, Airbnb, Google, Facebook, they're all using R to do data science on the data that they collect. Okay, and this is a survey, I think, from Australia, but so they found that employers are looking for this skill in the future job ads. Okay, so to summarize, it's free, it can handle large data very, very easily, it generates beautiful graphics, and you can develop reproducible analysis in R. Yeah, it's just a summary of all the things that I've just said. The other beautiful thing about R is that it does not mutate your data. So when you are reading the data in, it allows you to exactly define event. Let's say you have data in a table form, and there are some dates in that data column. There may be some just numbers or some words or long text. When you're reading that file in using R program, you can specify what format is what. So it completely rules out the chances of mutating the data. I don't know if anyone has across that some of the G names in Excel can be converted, Excel automatically will convert them into date format, and it just corrupts your data. And that has happened, and it has happened more often than you would imagine. So this is an ex-colleague, he's based here in Melbourne. He looked into existing databases. So these are, official repositories of bioinformatics data, and he picked up several errors that were introduced because people used Microsoft Excel to do some analysis on the data, and it introduced errors. Okay, so just be aware of that. If you're doing bioinformatics, I would say choose R over Microsoft Excel. Okay. Because I borrowed these slides from another course that I teach on R programming. So I have, some terminology that I had on my slides. I didn't remove it. So when you're talking about R, you would hear a lot about, this package, that package, what does that mean? What does code mean? What does syntax mean, or object? So these are the terms that you will commonly hear when you're, if you're new to R programming. And hopefully in the afternoon session, when you are writing your own R code, you will hear some of these terminology again. Okay, so what is a package? So package is like different apps. So, I'm sure everyone has a smartphone these days, and when you buy a phone, it comes with some preloaded app. So those are some of the basic apps. So you can imagine that when you're installing R for the first time, it comes with its own built-in packages. So those are some of the basic packages. But then you want to use some new apps with R. So those are the packages you will have to install. And Bioconductor is one such library of all those packages. Okay, and you can have other. There may be other community-specific projects where you will find some other specialized R packages. And normally, I teach students, what are different ways of installing an R package. Okay, so this is one of them that you can, if you are using RStudio, you would see that there is an option to install R package, and you can select where you want to fetch that package from, because there are, as I said, different designated repositories that are available where you can fetch this package from, and you can directly install it just with the click of a button. Next is code. So code or source code is the term that we use to, for a set of instructions. So if you are writing a R code, so you write some instruction, normally using English, letters on the keyboard and numbers and some symbols, and which then computer will interpret to do the task that you have asked it to do. Syntax is the next term. So syntax is just a language. So when we say English, there are some rules and convention to write English language. Similarly, R programming has its own rules and convention to write R programming. C programming will have its own syntax, and Java and so on. So every programming language has its own syntax, which is the set of rules. Objects are the variables that you use. For example, you say x equal to 12, y equal to 20, 2x plus y. So x and y are the placeholders for those values, and those are called objects. So just some very, very basic terminology for R. Okay. Okay, so just briefly on what RStudio is, if you haven't used RStudio before. So there are two ways to run R. One is through command line. So when you install R, it comes with its own console. So console is nothing but the command line version, and you can directly type a command on command line and it will execute that for you. Okay. So for example, in this example, we are doing, we are writing an R code to draw a histogram, and it will display that on the right-hand side here. Okay, but so what do you see as a disadvantage? If you had to run all your R code on command line, what could be some of the disadvantages? Anyone wants to share their experience?
You cannot automate many of the things.
Okay, you can't automate. Good one. Anyone else?
We can't see histograms, diagrams, representations.
Mm-hmm. So you will have to save the graphics that you're generating. Yeah. What else? And if you wanted to rerun this code, you should save it, right? So if you're running all the commands on command line, when you close it, it will disappear. Okay, so you can't save it as a file here. So you'll have to use an editor to write your commands in a file, and then you can reproduce that code. So for all of those reasons, you will move to some other additional application that you will use with command line to run codes. Okay, so this is called a console. That's the output. Now, this is how an RStudio window will look like. Okay, so if you don't have RStudio, I would say just today, download RStudio and open it and observe all of these different options in your RStudio window. So you will see these 4 sections when you open RStudio. So the top left section is the editor where you will write the code. So as I said, if you want to save this code to reuse later on, you will write this in an editor, okay? And this is the console that you were just now seeing. So it comes up. So on top of console, you have an editor. And on the right-hand side, you will see all your plots and files, etc. And it also has browser options from here to open a file or even export something. And then you can also install packages from here. Another very, very important feature in RStudio that I find is this environment and history. So every time you create a new object, new variable here, it keeps track of that variable on the right-hand side. And you know that variable has been created and what type of variable it is. So RStudio just makes this experience of learning R programming much more easier, because you're able to see all the things that are happening behind the scene. You know where your code is, and you can immediately see the code, and this is running on the console, and the output and history, all of that in just single view. Okay. Right, and you can resize all these 4 sections. Okay. So that was just a brief introduction to RStudio. And similarly, you have Google Collaboratory. I don't know if you have used it. If you're doing programming in Python, you may have used Jupyter Notebook, and Google came up with this browser-based Jupyter Notebook that you could just access by logging into your Gmail account and searching for this app called Collab. Okay. And again, you can run the code from inside this notebook, and it shows you the output here. And there are some existing tutorials that you can go through. If you are learning to program in Python, I would highly encourage go to their existing tutorial and try and reproduce those analysis. And you can also upload a new file here and can run the same analysis on your own data there. Okay. Okay. So with this, I'll take a 5-minute break, and I want you guys to open RStudio if you already have it on your computer, or download RStudio and open and observe, what you see. If you have any questions, let me know. Okay, so we'll just take exactly 5 minutes break. So this is 3:17 for me. So 3:22 I'll resume. Okay, so I would like you to look at RStudio or Google Collaboratory if you are doing Python programming and not R. All right, I'm back. So I hope you found RStudio. And sorry, earlier I didn't realize this is in a, Zoom is in a webinar mode, so it's not two-way conversation, but I now see the participants have talking enabled, so that's good. Okay, so did anyone look at RStudio?
Yes.
Yeah, do you see 4 windows like this?
Yeah.
Good. So this is what I was talking about, that you can type your code here, and when you execute, you will see the code is executed here in your console. And when I create a new variable, the variable goes here in the history. Okay, so now I'm creating another variable y, and that goes in the history, and it's done. And so if I do x plus y, oops, it will give me the result here. Okay, so yeah, if you haven't, try running some code, and you can save this. So you can save this file as a, I can save it on my desktop, whatever, something R, and then this code will stay for me to reuse. Okay, yeah, so I would just say, try out some existing R code, and I'm sure you'll be learning one R package this afternoon. So try and do more practice with this tool. Okay, I'll move back to my slides. Are there any Python programmers in the audience?
Yes.
Yes, ma'am.
Good, good, good. So did you try Google Collaborator before?
Yes, ma'am.
Yeah.
Beautiful. Good. Sorry, I lost my slide. I'm going back to that now. Okay. So if I go back to my Zoom. Okay, should be able to see this now. You see the full screen again?
Yes.
Cool. Okay, yeah. So try this. Now, let's look at some bioinformatics related stuff now. So who has heard of the term NGS? Sequencing in general?
Yes. Next
Okay.
generation sequencing.
Next generation sequencing. Well done. You want to share what you mean by, what is your definition of NGS?
So my definition of NGS would be getting any sequencing data out using highly parallel methods,
Beautiful.
going beyond traditional microarray methods.
Mm-hmm. Okay.
Yeah.
So can I assume everyone is aware of what sequencing, DNA sequencing is? I don't know, probably in your course you would have studied Sanger sequencing and then next generation sequencing, yeah? So, you know the rule of complementarity in a DNA sequence. So G always pairs with C and A pairs with T, yeah? So with that rule of complementarity, if you know the sequence of a DNA strand, you are able to write down the sequence of its complementary strand, okay? So this is how Sanger sequencing technique works, and the same is the basis of all of these high throughput sequencing technology. So the basics are the same, that you have a template and you are resynthesizing its complementary strand. So in high throughput sequencing, as the name suggests, you're doing this over and over again on a very, very large scale. Okay, so if you get only like 2 or 3 copies of a sequence in Sanger sequencing, here you're getting millions of copies. Okay, and because you're able to get millions of copies, of a single piece of DNA, you are able to do more samples and more coverage of your sequence. Okay. Now, I personally have reservations about using the term next generation sequencing because next generation came after, the low throughput sequencing, which was Sanger. So I don't think this term holds true anymore because now we are into third generation of sequencing, which is real-time sequencing. Okay, so I like to use the term high throughput sequencing or massive parallel sequencing, but NGS has been a very popular buzzword for a long time. So you will see this everywhere. So this is what it means, high throughput sequencing. Okay, when you're sequencing something, so you will have a biological sample and you will sequence it on some magical sequencer, and what you get out is its DNA sequence written, read, all of the letters and their order recognized. Okay, now current limitation of these technology was that it can sequence only a certain length of fragment. Okay, so if you have thousands of base pairs in your DNA sample, you'll have to break that into smaller pieces. So normally these short read sequencing technology, they require those fragments to be broken into, let's say, 500 base pair, and then it will sequence it. So what you get at the end is lots of broken pieces of your original sample. Okay, and after you have sequenced a sample, you'll have to stitch those together yourself and rebuild the original molecule. Okay, and that's where the skill of bioinformatics comes into picture. Okay, so we have this data generated for us, but it's in a format that cannot be read. You can't find any biological meaning out of these lots and millions and billions of these tiny pieces. Okay, so in this group of sequencer, what you get is short read sequencing, and that read length. So read is the part that has come out of this machine. Okay, and the length of this short read sequence is between 50 base pair to 250 or 300 base pair. Okay, and now you'll have to, your aim will be to put this all together and make longer stretches then that can be studied for biological information. Okay, so after this point, your bioinformatics task begins when you'll be doing data analysis. Okay, and that's where you need all these skills. You need skills in programming, you need skills in statistics, you need skills in some computational algorithms, and obviously, very, very importantly, you need biological knowledge, so you know what you're looking for, and when you generate some results from this data, you are able to make sense of that. Okay, so my screen looks a bit funny here, but have you heard of these standard sequence data format? So FASTA, has anyone heard of a FASTA format before?
Yes.
Yes, ma'am.
Good. I'm happy to hear some voices. I can't see you guys, but good to hear that you're here with me. I'm not just talking to my computer screen. Well done. So
hi, Sonika.
How does a FASTA format look like? If you have to explain.
Hi, Sonika. So there was a one full day session on NGS that the students participants had.
Okay.
FASTA, FASTQ, the QC, and then data.
Thank you for letting me know.
Yeah.
Thank you for letting me know. I can quickly skim through this then.
Yeah.
Awesome. Beautiful. No, thanks. Yeah, please intervene and let me know. Okay, so just to recap, FASTQ has all this information, and have they done quality control and looking at the quality string as well, Raghu?
Yeah, they did. The one session was on QC of and understanding the FASTQ files. Yeah.
I'll then completely skip this section.
Okay.
Okay, so just to remind you, there's an ID in your FASTQ file, there is a sequence, there's an empty optional line, and then there is the quality string, okay? And each of these letters mean some Phred quality score that varies between 0 and 40, and which can be translated into an error probability or an accuracy value. Okay, so the quality of data is very, very important, as you may have already heard. And it's very important to know what sort of quality scheme the data was generated with. Okay, so as you know, there is a different set of quality letters that are used by different versions of these sequencing software. Okay, as I said, I'll skip through this. Yeah. So I'm sure you're familiar with these plots, the FastQC plots, and what we are aiming for is to get data that is above this red zone here, and it's of reasonable quality that you can be sure that the downstream analysis will be good. Okay, sequence alignment. Have they studied sequence alignment?
Yeah, they are familiar with sequence alignments. Yeah.
Okay, cool. So just again, very briefly to recap, so sequence alignment is you have all these short read data that's coming out of sequencer, and then you want to align this with a reference genome sequence, okay, if that's available, or any reference sequence. It could be a transcriptome sequence, okay. So, and when you align this, you allow for mismatches because sequences have errors and there are some natural mutations in the biological sample. So you want to account for those when you're aligning the data. Now, when you're choosing an aligner to align your data, you need to keep in mind what is the purpose of your alignment and what is the kind of data that you're aligning. Okay, so it may be whole genome sequence data, or it has been resequenced and you are matching it with another genome sequence. Or you could have RNA-seq data. So in case of RNA-seq, you are sequencing the messenger RNA or the entire RNA content of a cell. And what you will get is the transcripts that have been sequenced. And when you want to align those transcripts back to genome, you need to remember, where this data should align. Okay, so let's say if I was only sequencing messenger RNA, and when I align it back to the genome, then I expect that all the genic, I should see alignment in all the genic regions, or exons to be specific, because it's normally the spliced RNA that you're sequencing. In case of ChIP-seq, you are sequencing a piece of DNA that binds to protein. And again, when you align it back, you will only see patches of alignment throughout the genome. Okay. And so there are various, when you're running an alignment, you have to remember what my input data was, what reference I should use. Do I have a reference from the genome that I got the biological sample from? How many mismatches does my aligner allow by default? Is that enough for me, or should I account for more, or should I not account for any errors? So all of those things you need to ask all those questions when you're starting your alignment process. The other important thing is every sequencing platform may have its specific error profile. Is the aligner handling that? Because aligner was developed as a generic software, and you may have to sometime fine tune their parameters. So all these things are important to keep in mind. So you don't just blindly follow any recipe that you found on the internet or in the tool manual. Okay, always ask the questions. Does my are these requirements fit to my data type and the biological question that I have? Okay, so it's never one size fits all kind of solution. You always have to customize these tools to suit your need. I already said these things. And another way to identify what tool might suit my need is to just maybe generate a matrix like that. And what I have here is the name of the tools, what algorithm they are based on. This is important because it will decide what sort of computer infrastructure they may need. Can I run it on a standard sort of computer station, or do I need access to a high performance computing cluster? Does it support long read or short read? Does it do gapped alignment or not? Can it handle paired read? Can it do spliced alignment for me if I was doing RNA-seq? Okay. When you do an alignment, you get a SAM format, and then SAM can be converted into BAM. And again, I'm going to skip through this because you've already covered the alignment part from here. So SAM stands for sequencing and alignment format, and it's a text format. And then BAM is a binary format, which reduces the size of this format by 1/3 or 1/5. And then there are tools that can directly operate on a BAM format. And you can find aligned files available in the public repositories there in BAM format. Okay. So you may have already seen, this is a typical, SAM format output. It has these 11 columns, and what those columns mean, again, I think I can skip this safely. If I have to explain this, please, yell out. Okay, so, and as I said, this is one of the tools. Again, this visual is not looking very good, but this is an example of an IGV browser where you can import a BAM file and visualize it. Okay, from here, we will go into RNA-seq topic. Okay, now that you already have some background to what sequencing technology is, you already done quality control, you already done alignment. What I want to see from you guys is to look at these keywords on my keyboard, on my screen, and try and arrange them in an order of a workflow. Okay, so if these were the key steps of your RNA-seq data analysis, what comes first? Okay, so try and arrange those keywords into an order. So I'll give you 2 minutes to think about, and then if I can get some volunteers to give answers, that will be great as well. Okay, anyone wants to give it a try? Maybe first few steps. What do you think? Which of these labels I should put first if I have to write down a workflow for RNA-seq data analysis or an RNA-seq study? Sorry?
Experimental design would be the first, and maybe detect.
I couldn't hear you. Experiment design, yeah.
Yes.
Experiment design, and
then it might be library preparation, and then sample raw Illumina RNA FastQ. Then you would be having assembly. After that, you have indexed means so sample transcript assembly, sample ungapped alignment, and sample spliced alignment, and then you have indexed reference genome FASTA. Sorry, first you have, so let's go with the experimental design. Then you have library preparation, then you have sample raw Illumina RNA-seq FastQ, and then I'm missing the experimental steps. But yes, after once you have the indexed reference genome FASTA, then you would be doing sample ungapped alignment, then sample spliced alignment, and then a quality check at every step, and yes, detect differential transcript expression at the last.
Okay. Anyone else? Good, good, good. I like your answers.
I agree with him. Just I think that the quality control would be with FASTQ file, and then we will convert it into index reference genome FASTA file.
Okay, good one, good one. I like that correction. Anyone else? Okay, no more volunteers, so we'll move on. So this is how I would put this all together. So obviously all this comes before you start doing any analysis. So experimental design is very, very important. Okay, when you're getting, even if as a bioinformatician, if you're getting the data to analyze, you must always ask what the experiment design was, how the libraries were prepared, and how the sequencing was done, because all of these steps are very, very important to decide the parameters for our downstream analysis. Okay, I like what you said about QC. There should be QC at every step. I really like that comment, and definitely you have to have QC right in the beginning, but then at every step we should have QC. Okay, and yeah, I like that correction that the index genome will come here, because to do any, I don't know if you covered that in your previous talk or hands-on session, you'll have to index the genome with all of these aligners, because they're handling such large data that require genome to be indexed first before you can start using it. So that's a step in the analysis. Okay, so along with the row sequence reads, you will also give a reference index genome as an input, and then you will start aligning. Okay, and because we have RNA-seq data, and I'm assuming, again, I made an assumption that this is eukaryote data, so it's this spliced alignment we are after. And normally in the splice alignment, there are two steps that the aligner will first just do an ungapped alignment, and then we'll find the splice junctions and then report the spliced alignment. And then from there, you can either directly go into differential expression analysis by doing like looking at the aligned files and counting reads that are aligned to each gene, which is called gene quantification step, or you want to stitch all these alignments together to build complete transcript sequences. Okay, so this assembly step is optional, but it becomes very, very important when you're studying a genome where assembly is not perfect or not completely available. Okay, so you will first do an assembly step and then perform the differential expression analysis. Okay, but there'll be lots of variation to this workflow, but roughly these are the steps you will follow for a eukaryotic RNA-seq data analysis. Anyone has a comment or a question?
Just one question, ma'am. So let's say you have a transcriptome. So would you be aligning it to the genome or the transcriptome of Homo sapiens? So let's say we are dealing with the Homo sapiens data.
Yeah, so it goes back to the point I was making earlier, that you have to, when you're choosing a reference, and then you have to ask this question exactly, that what is it I should use as my reference? Should I go for genome or should I go for transcriptome? So my answer is, if you have a well-studied, well-annotated genome, use genome as a reference. But if you have some special requirement, some special question that you're answering for transcriptome from your experiment, you may have to align with transcriptome, even if genome is available. But generally, my answer will be that if you have a well annotated complete genome, go for genome. And if you don't have genome and only have RNA-seq data available, then you will align with transcriptome. And in some cases, you will first build a reference and then align your reads to that for getting counts for each group that you have in your experiment. Any other questions? But yeah, as I said, you ask those questions, like what are the requirements? What has my experiment generated? Because you can only capture the biology that has been, captured in your experiment. Okay, so you need to make sure what your limitations are and what information I'm expecting in my data, and then what is the question that I'm trying to answer downstream. Okay, so you may be doing RNA-seq analysis on samples from different kinds. You may have sample from an organism, from cancer, plant, could have data from soil, or you could have RNA extracted from any of these biological samples, and then you want to quantify. So you want to see which gene is expressed and into what quantity, and then you may be comparing conditions. For example, you're comparing melanoma with a normal tissue, wheat in 2 different conditions, and so on and so forth. Okay, so RNA-seq is the technique that you can use to do a dynamic range of gene expression, and you don't need to know the gene annotation beforehand. You can work it all out from the data itself if you don't have that information available. So experiment design, as I said, is very, very important, okay? And you need to make sure you spend a lot of time in planning your experiment. How many samples you need to run, how many replicates you need to run, what type of sequencing, how much sequencing you need to do. So there are lots of, guide available, various papers available on, how these things, how you can decide on these things. And again, for a statistically viable experiment, you need to run a minimum number of samples and do a minimum sequencing depth. Okay, without that, there is no point performing a gene expression, differential expression analysis from more than one condition. So something happened to my slides. It's not streaming properly. Okay, but what I was trying to show in this case is that when you are, you may have learned about the multiplexing technique. So if you were doing sequencing like on Illumina, it has multiple lanes and you can load multiple samples on each lane. Okay, so I would prefer design on the left than on the right. So for example, if you have these 6 samples represented by 6 different colors, so don't just load 1 sample per lane. Try and mix that equal amount and have equally distribute them across lanes. So that will minimize the between lane effect on your final data. Okay, so those kind of things. And again, for sequencing, they will get evenly sequenced, because each lane has this fixed sequencing number of sequences that it will generate. Another is a replicate versus depth. So do as many replicates as you can. And normally, 3 is the bare minimum that you would do, though, statisticians don't like that number, but they want to see at least, 10 or so replicates generated. But, extra sequencing also costs money. And if you have to make that trade-off, so, do lots of replicates and then replicates versus depth. So depth will determine how, how precisely you can get the, how much resolution you can get in your analysis. So depending on that, you will also decide on depth. If you're only comparing between 2 conditions, then you can go with the minimum depth. But if you were going to resolve between different, let's say, splicing variants, then you want to go deeper in the depth. Okay, so next was library preparation. So library, this is a better slide to show you. So this is the cartoon of an RNA content in a cell. So you have various kind of RNA in the cell. And you can see in this cartoon, the majority type of RNA is the ribosomal RNA. And then there are messenger RNA, and then everything else is represented by this blue color. And normally majority of the RNA-seq experiments are done to capture this messenger RNA or the protein coding gene, okay? And we're not interested in capturing ribosomal RNA. So there are 2 ways to prepare RNA from here. One is to do poly-A selection. So normally these messenger RNA have a poly-A tail, so you will select for poly-A, and you get all the messenger RNA selected, and you sequence them, and you get your coding gene sequence available. The other way to do is that you remove all the ribosomal RNA, just subtract that, and what you are left with is messenger RNA and everything else, okay? So this is called the whole RNA sequencing. This is called messenger RNA sequencing. And again, depending on what your interest is, it's just the coding sequence or the coding and non-coding RNA, you will choose between these 2 protocols. And again, none of these captures are like 100% perfect. So when you do this capture, you expect some other type of RNA that creep in. Similarly, when you do ribo-minus, you would still see some ribosomal RNA coming through in your final data. And again, these are just part of the experiment, and you are aware of those. You will look out for any signs that will show you that there is some, you only want to study coding, but there are some other transcripts in my data, and how you want to handle those. Okay, then you do sequencing and you do QC. And what we have here is the next step after QC. So you will align. So this was my original gene, and the reads will align at different places, because what we are doing is messenger RNA sequencing. Then I get bits and pieces of this messenger RNA sequenced, and this is how the data will align back. Okay, there are different aligners. Sorry, there was a reference here. There are different aligners that are available to map RNA-seq data. There are actually two new ones that, one new one, HiSET2, can be added to this list. Okay, and they have done various benchmarking between these aligners, how well they perform. Okay, and then after you've done the alignment, so this is an example output from TopHat. So what you get is a SAM file, and then again, you should, you have already covered this, but you should be aware of, you know, what the information in my SAM file, and if there are any investigation you need to do, you should be completely across this SAM format. So if you haven't done so, I would highly recommend read the SAM manual and understand what each of these columns mean, what these different symbols mean. And this is very, very important, especially if you're doing RNA-seq data analysis or variant analysis. Okay, after you have the BAM file, so BAM file is like, you know, you have a, you know, pin on your Google map, and then you want to tell, you know, what is it, what is available at this location, what is the building like, and then you add more specific information to that. So BAM file has all the location of your reads onto the genome, and then you say, oh, this from here to here, these reads belong to gene X or gene Y, and that's how you count. You start binning those aligned reads into different gene locations, and that's called the gene quantification step. Okay, again, that read summary can be done with assembly, for example, using tool like StringTie, or it can be done just directly counting number of reads aligning to each gene. And when you have done that, what you get in the end is a table like that. You have genes as your rows and your samples as your column variables. Okay, so you have gene X has these many reads from each sample, or this is called the expression of that gene, digital gene expression table. Okay, so after you have converted your RNA-seq data into these gene expression table, then you can do different analysis. So for example, you have this table for the wild type group and the condition group, and then you want to compare, okay, whether the expression of this gene is any statistically different between wild type group and the knockout group in this case. Okay, now to do this, there are various tools available. One of them is EdgeR. Do I have that? Yeah, EdgeR, Womb, DSeq. So all of these three tools that I have commonly used to run RNA-seq differential expression analysis, these are all available through Bioconductor. And you can, it requires inputs like this, that you have a gene expression table, and these should be raw counts. You should not have performed any other operation on this. Don't normalize this data before you input into EdgeR. Okay, these are just the direct counts that were summarized from a BAM file. Okay, all right, so now, during the differential expression analysis that you'll be doing a normalization step to normalize for the difference in sequencing depth for each sample. And also if there are any outliers in your data, all of that will be adjusted before you start comparing the expression between one condition and the second condition. So a normalization is done so that you are only comparing the biology between the two groups and not any technical noise, okay? And there's some extra information on my slides. I'm happy to give a copy of these slides for you to read later on. Something to do probably with my internet speed here, or I don't know, the streaming is not perfect. Okay, so in this case, so these are the things that you need to be aware of. So for example, if gene E, so it has expression of gene A in 3 replicates, and I don't know, I can't really explain this without this proper table. So sometime you will see that one of the replicates has an expression value which is very, very different to the rest of the genes. And this is where in such cases, you want to have an approach, a statistical approach that's robust to this kind of scenario, and it correctly corrects for this kind of differences. And in such cases, having extra replicates help. Okay, so if one of the replicates didn't behave out of 3, then you don't have enough replication in your study. So that's why they say, do more than 3 if you could, because if you have to take out one of the replicate for these regions, you still have enough data to perform your statistical analysis. So yeah, going back to that, our experiment design question on, how many replicates you should have. The other concept is of multiple testing. So if you're doing any differential expression analysis, you will have thousands of genes that you're testing. Okay, so you have thousands of testing hypotheses that you have to perform between 2 groups. And so any statistical test you are performing to compare the expression between 2 groups, it will give a p-value. Okay, so it's very, very important when you're doing RNA-seq analysis that you correct for false discovery rate, which is called multiple testing hypothesis, and do not report just p-value as it is from the statistical package, but report its FDR value alongside, so that you have corrected for those false discovery rate effect. And there are, again, all these packages give you different options to correct for that p-value. Okay, yeah. Now, at the end of differential expression analysis, what you will have is list of genes that are changed between 2 conditions, and with the, like, by what extent they have changed. So for example, you will have, gene X is changing 2-fold between 1 condition and 2nd condition, and then you want to see whether its p-value is significant or not. And by setting a cutoff for p-value, you have a list of statistically significant differentially expressed genes between 2 conditions. Now you only have a list of genes. You'll have to make sense of those genes, and you want to see are these genes involved in a particular type of biological process or pathways. And that is the next step, to add biology to your list of genes, and you will do biological interpretation. And again, there are a number of tools that are available, David being one of the oldest tools that has been around for a while, since the time of microarray. And you can just paste your list of significant genes here, and it will do a mapping and will show you whether there are any special biological pathways that are coming up as overrepresented in your list. Okay, so there is also a consortium that were doing the systematic analysis of various RNA-seq protocols and RNA-seq library preparation types and sequencing platforms, to give you an idea, what is how the performance between different, experiment design methods as well as sequencing methods. Okay, with that, I would like to thank you all for your attention, and happy to take any questions.
Thanks, Sonika. That was a very interactive talk. I have one question. So most of the time we come across tools like, let's say, 5 years down the line, and we are not able to even get access or find out that the tool that we wanted to use doesn't exist, and then we are scrambling to find a new alternative. So what has been, in your experience, that you have done to overcome this type of scenario?
Sure. Yeah. So I think, yeah, it's like an open question, and also something I don't think I can get a direct answer to either. So again, when you are deciding on what tool you want to use, you want to go with what is the best community practice for that kind of analysis, because I don't think there's a set rule that this is the tool you should use. So there are always 3 or 4 methods that are very popular in a particular domain of analysis. And it does happen that tools are not maintained throughout. For whatever reason, funding runs out, people move on to different jobs. So it has happened that even the same tool that I have used previously, I had to move on to using another tool which does similar things. So that's why when I'm teaching students, I tell them, try and understand the concept. What is it the tool is trying to do? Okay, so it's not just that tool will do your analysis. If you understand what is it the tool was doing, what type of alignment I wanted to do, for example, is there another tool that gives me all of those properties? And then it's easy to make a switch and easy to make a selection of another tool that does same kind of job. But it's been a problem. There are so many tools. There are probably an aligner published every month or maybe every 3 months, and there are too many aligners out there, and not everything is maintained throughout its life.
Thank you. Anyone else has any questions? Please unmute and you can ask.
Okay, so let me know, Raghu. I hope this was useful and it will help you guys for your afternoon session as well.
Sure. I think since some of the participants are from biotech background, this would have been useful for them and would be a good kind of like basics things that they would like to go ahead with the next few sessions that's that for today.
Mm-hmm.
Yeah.
Mm-hmm. Okay, good to hear.
Okay, does anyone have any questions? I don't see much on chat or the Q&A. Yeah, please go ahead.
Dr. Tyagi.
Mm-hmm.
Yeah. So I have a question. It's a kind of a very a simple one. So when you are choosing, when I had to machine learning, so how can I choose what is the kind of programming language I can use? So we have packages with R. Also we have packages with Python, right?
Yeah.
So what are
Okay.
the things I have basics that I have to see?
Yeah. See, this is up to you. What is it that you're most comfortable, doing programming in? So as far as R and Python are concerned, there is active development happening in both the programming field, right? But whatever language you pick, go in the depth. Okay, so if you know only a little bit of this and a little bit of that, then you can't use the full capacity or the full power of that programming language. So pick one and then go into the depth of it. But as in bioinformatics field, both programmers are equally successful because there are tools and methods available for both the programming languages. So in the beginning, there were more development happening for machine learning packages in Python. Everyone in my lab does Python programming for that reason, but now I'm seeing this more like same package, like for every Python package, there is an R package available.
Okay.
But it's up to you. What is it that you're most comfortable with?
Okay. Thank you.
And there may be another language. I think Ruby is picking up now for natural language processing. So who knows? These things will keep changing.
Yeah, and then there's Julia as well. So
And
that's
Julia,
also
yeah.
there's picking a lot of speed in with Julia.
Yeah.
So
yeah.
So it is always a good practice to have an eye on what is happening in the development field of both of the packages, both with respect to R and Python, right?
Yeah.
Yep. Thank you.
anyone else has any questions?
If you want to type in, if you're shy to speak up, feel free to type in. That's fine.
Okay. Okay. So just to reiterate, you can always contact the speaker through email, and they would be able to respond to your queries, which has been something that all the speakers in this workshop have been mentioning. So in case you are getting a question later on during the workshop or after the workshop, feel free to ask them. I think right now we can end this session. So I'd like to thank Dr. Sonika Tyagi again for taking her time out for giving this presentation and participating in this workshop and talking to participants about the tools and the open source tools for genomics and about her work that she's been doing at Monash. So thank you once again, and we look forward to having you at some other time in the future.
Pleasure. Thanks for having me here.
Thank you, Sonia.
Yeah.
Thank you. Have a good day. Bye. Okay, so there's an announcement for all the participants. So after this short break, we will continue the session 2. So you will have to log in using your Windows ID for the session 2. Okay, we'll pass on this message in the WhatsApp group as well. Okay, we'll see you after a break.
So I'm going to use our
Is recording stop? Live? Stop.